A 68-year-old man arrives in the intensive care unit. For 2 days, he's been lying completely immobilized on the floor of his home after a collapse. His right leg, pinned beneath his own body weight, is swollen and tense. His vital signs are rapidly deteriorating. His blood pressure is dropping, and worst of all, he is barely producing any urine. What little his kidneys do manage to push out is a dark, alarming tea color. The medical team runs a rapid point-of-care test. The chemical reagent strip lights up instantly, screaming a severe 4+ positive for blood in the urine. But when the lab puts that exact same sample under a microscope, they find absolutely 0 red blood cells. To confirm, they spin down a sample of his blood in a centrifuge. If he were suffering from massive internal bleeding or destroying his red blood cells, the separated plasma would be pink or red. Instead, it is perfectly clear. The test result is a chemical illusion. The reagent pad isn't reacting to whole blood. It's reacting to an invisible rogue molecule that has bypassed standard detection, hiding in the clear plasma while systematically tearing the patient's kidneys apart. That molecule is myoglobin. Normally, this tiny 17 kilodalton protein is locked safely away inside the cytosol of healthy skeletal and cardiac muscle cells. In its proper environment, myoglobin is essential for human endurance. It acts as an intracellular oxygen storage tank with a massive affinity for oxygen, holding onto it tightly and only releasing it to keep muscle fibers alive during intense exertion when local oxygen levels plummet. But the patient's prolonged immobility triggered a cascade of mechanical trauma. 48 hours of constant crushing pressure physically tore open the cellular membranes of the muscles in his right leg, a condition known as severe rhabdomyolysis. Through those torn membranes, the ruptured muscle fibers dumped their massive reserves of myoglobin directly into the surrounding tissue and the systemic bloodstream. A specialized protein, perfectly engineered to keep cells breathing in a highly specific, localized environment, has just been unleashed into the general circulation as a lethal biological weapon. Because myoglobin is so incredibly small, it slips easily through the kidney's initial filtration barrier. It moves straight out of the blood and directly into the delicate, microscopic tubules responsible for processing urine. Once inside, it enters a hostile environment. Because our patient has been lying on the floor without water for 2 days, he is severely hypovolemic. His internal fluid volume is low, making his urine highly concentrated and dangerously acidic. As the myoglobin travels deeper into the nephron, reaching the distal convoluted tubule, it encounters a normal structural molecule called TAM-Horsfall protein. Under normal conditions, they would pass each other by, but in this highly acidic, concentrated state, the myoglobin and TAM-Horsfall proteins aggressively lock together. They precipitate out of the fluid, forming dense, dark, solid masses known as casts. These hardened casts act like biological concrete. They physically choke the kidney's microscopic plumbing, creating a massive backlog of pressure that violently halts the organ's ability to filter. This creates a catastrophic mechanical failure, abruptly stopping the body from clearing its own toxic waste. While the distal tubules are filling with concrete, the cells lining the proximal tubules, further upstream, try to manage the crisis. They actively absorb the excess myoglobin molecules from the fluid into their own cellular bodies. Bringing myoglobin inside the cell sets off a bomb. The acidic intracellular environment forces the myoglobin molecule to destabilize and break apart, shedding its central iron core. This unleashes free ferrous iron. The iron immediately reacts with ambient water and oxygen molecules, which sparks the Fenton reaction. This chemical process generates a storm of highly reactive, unstable molecules called hydroxyl radicals. These radicals rip through the cell's internal architecture, triggering severe lipid peroxidation. They shred the mitochondria and force the tubular cells to undergo apoptosis, effectively burning the kidney's tissue from the inside out. At the exact same time, a third attack is underway. The broken myoglobin molecules circulating nearby act like chemical sponges, scavenging and depleting all the nitric oxide from the kidney's surrounding blood vessels. Nitric oxide is what keeps those vessels open and relaxed. Without it, the renal arteries violently spasm and clamp shut. The patient's kidney faces a horrific triple threat: plugged with biological concrete, burning from a chemical fire, and suffocating without a blood supply. Because myoglobin is so small, it floods the bloodstream immediately after the muscle ruptures, peaks within hours, and is rapidly flushed into the urine. By the time a patient actually arrives at the hospital a day or two later, the myoglobin levels in the blood have often already plummeted. This chart shows the timeline of biomarkers in the blood following muscle trauma. The first line, myoglobin, spikes aggressively in the first few hours and then rapidly drops to zero. Because it vanishes so quickly, physicians rarely catch it in the act. Instead, they look for the smoke it leaves behind, creatine or CK. This second, thicker line shows how that much larger muscle enzyme leaks into the blood slowly and stays elevated for days. Once those massive CK levels confirm severe rhabdomyolysis, the medical team initiates the primary countermeasure, high-volume intravenous fluid resuscitation. They flood the patient's systemic circulation with isotonic crystalloids. The goal here is to push fluid through the renal system at a high velocity, mechanically diluting the concentration of myoglobin inside the nephrons. This sheer volume of fluid acts like a biological fire hose. It raises the flow rate and decreases the acidity of the urine, preventing the myoglobin from binding to the TAM-Horsfall proteins and physically forcing the forming concrete out of the system. The patient's survival relies entirely on the critical care team recognizing the dark, dual nature of the myoglobin molecule. In its proper context, myoglobin is an evolutionary triumph. It is the quiet, highly efficient mechanism that allows human muscle to sustain extreme physical endurance. But the very traits that make it so effective at gripping oxygen and surviving inside the muscle cell turn it into a relentless engine of destruction the moment that cellular boundary fails. The human body relies on incredibly precise compartmentalization to function. The chemical tools that keep our tissues alive are entirely dependent on their geography, perfectly safe and absolutely vital,